There will be pizza.
For my seminar
Seminar,
presentation.
okay. Did everybody get the email I sent that had the revised list of tests for your unknown and the study guide? Okay. Like I say, I know you don't want to study over break, but it wouldn't be a bad idea to get some of the lab exam stuff. Just to kind of keep that fresh in your memory. Okay? Hang with me for an hour?
Don't give me guarantees, but.
No, okay. Well, you don't have a note from Mom, so you've got to say.
Yeah, I know, right?
Okay, so we're in the midst right now of going through the second line of defense, and remember that the second line of defense involves a pretty complex interaction between some cellular components and various biomolecules that cells produce. The cells that produce these biomolecules can be pathogens, but a lot of the cells that produce them are also cells of your body. Some of them intimately associated with the immune system, but other of those are more kind of structural components of the body. We mentioned epithelial cells and fibroblasts, for example, that produce that interferon molecule, which is one of the chemicals that's involved in your second line of defense. We just started talking about the complement cascade, and the complement cascade is another very important part of your second line of defense against infection. What does complement consist of? What kind of molecules are we talking about?
Serum proteins.
Serum proteins, and there are 26 of them at least that we've identified to date. What produces them? Liver and white blood cells produce those serum proteins, okay? And they're constantly producing them, okay? So these things are constantly present in your blood, but they're present in an inactive state. And what has to happen for the complement cascade to be initiated is some kind of an event. And that event typically involves a pathogen infecting the tissues. I mentioned that there are 3 different pathways for the complement cascade. There's the classical, which we'll spend the most time talking about. There's an alternative or properidin pathway, and then finally the lectin pathway. All 3 of those pathways converge minimally, at least at the same point, and that is they result in the formation of an attack complex. And the attack complex will punch holes in the membranes of cells or envelope virus. Any idea which type of bacteria, gram-positive or gram-negative, might be most prone to being destroyed by complement? Okay, gram-positive or gram-negative? Gram-negative. In general, gram-negative bacteria are more susceptible to complement lysis. Why? Because in gram-positive, you don't have that LPS outer layer. Complement can destroy that. And then they've got this big, thick peptidoglycan layer that kind of protects the inner cell membrane. So gram-positive, because inside, are not particularly susceptible to this pathway. What we're going to see in the classical pathway is that as you go from these 26 different separate proteins, and they interact with one another in this kind of avalanche type of set of reactions, where the product of one reaction becomes an enzyme that catalyzes another reaction, that along the way in the classical pathway, some molecules are spun off that have other factors and other functions. For example, some of those byproducts of the classical pathway, while the attack complex is forming, are factors that stimulate fever. Those particular compounds are called pyrogens. We'll talk about pyrogens in a minute. Some of them trigger inflammation. We haven't talked about inflammation yet, but that's a very important part of your second line of defense on immune response. Some of these particular compounds are chemotactic in nature. And chemotaxis is an important part of the inflammatory response. Remember last time we talked about diapetesis? We said that the white blood cells can crawl out of the circulatory system into the surrounding tissues, but there has to be something to attract them, okay? Another thing that happens during that process of diapetesis is molecules produced by inflammation start making subtle changes in the walls of the capillaries. And they grab those white blood cells more regularly. And we start getting that stickiness that occurs that allows eventually those white blood cells to attach to the endothelial cell wall and then crawl out of that tube, if you like, into the surrounding tissues. Some complement cascade byproducts are opsonins. I can't remember if I used that term previously. Did you recognize it? Some say yes, some say no. Opsonins are substances that enhance phagocytosis. There are some antibodies that can be opsonins, but there are also some complement proteins that can be opsonins. Well, what are these? Well, I don't know if this is still the case, but at least when my kids were growing up and when I was growing up, especially in junior high, you know, the joke used to be to put that sign on somebody's back that says, "Kick me," and then as you walk down the hall, people kick you. Opsonins are kind of like the kick me sign, okay? They are molecules that are attached to the surface of pathogens that mark them for abuse. And what's going to do the abusing? A phagocytic cell oftentimes. So by putting this kick me sign on the pathogen, it enhances the likelihood that that particular pathogen will recognize and then attach to and engulf that particular pathogen. Well, this slide is kind of a nightmare, and fortunately for you, there's only some general kind of take-home messages you need to take from this particular slide. So here we've got a cell that ultimately is going to be the target of this particular defensive system. This is a pathogen, okay? And ultimately what's going to result is that all of these little attack complexes are going to form on the surface of that cell. The attack complex are various complement components that have come together and interacted to one another to form this kind of channel in the membrane of the cell, or the envelope of the virus. What initiates this classical pathway? An antibody-antigen reaction. So that is the initiating event that triggers this particular cascade event to occur. We haven't talked about antibodies yet, okay? But one of the things that you should know about antibodies is that they are specific for particular partner molecules we call antigens. And if the antigens are foreign, i.e. they belong to a pathogen, then when that antibody-antigen reaction takes place, it's going to trigger some events that will ultimately and hopefully result in the destruction of the pathogen. And the complement cascade is one of the ways in which this can happen. There is really kind of a misunderstanding by the general public about how antibodies work and what they really do. Most people think that when an antibody is produced and it binds to a pathogen, it almost kind of like strangles it and directly kills it. Anybody ever seen, I'm sure you probably haven't, there's an old classic sci-fi movie called The Fantastic Voyage. And they reduce a submarine and some doctors into microscopic size, and they inject it into the blood of a person. There's a big blood clot. They have to go through the blood and blast away the blood clot with lasers and things. And at one point, they have to go outside to repair their little vessel, and the antibodies attack them. And they literally grab onto them, and it's almost like ropes around them, kind of strangling them. And that's what people kind of think is the idea of how antibodies work. No. Antibodies typically are just marking that particular cell for destruction. And one of the destructive mechanisms is the initiation of this complement cascade, resulting in cell lysis. What happens if you get a mismatched blood transfusion? Yeah, and it triggers an immune reaction, and the antibodies are reacting to the foreign red blood cell antigens. And how do you tell if that's happened in blood type? Do you remember? You mix your blood with various antibody A, antibody B. How do you know if there's a reaction? How do you know which antigens you have?
Coagulation.
Yeah, it kind of coagulates. It's called agglutination. It's called clumping. And most people think, well, that's the problem in a transfusion is your blood cells all clump together, and then the blood gets kind of thick and gooey and can't really do what it's supposed to do. Well, in reality, what's happening is in a mismatched blood transfusion, that your red blood cells are attached to by antibodies. But what does that do? That stimulates the complement cascade and results in red blood cell lysis. And that's actually the reason why a mismatched transfusion is lethal, is the lysis of the red blood cells. Not them all clumped together, the blood getting all thick and gooey. So what this particular slide shows you is just some of the nuances of the complement cascade. And if you were taking a medical school class or a graduate level biochem class or something, then you probably would have to memorize all these particular interactions and know that, this complement fragment C2 interacts with C4 to form something else. I'm not worried about that level of detail. What I am interested in you understanding is that the cascade results in all of these interactions taking place, and many of these products are enzymes that catalyze further reactions that have some outcomes. And one of the outcomes is formation of this attack complex. That's what lyses the cell. But other outcomes in the classical pathway are we spin off some waste products, if you like, that have these particular functions that are chemotactic in nature or stimulate fever. And all of these kind of coordinate together in different ways to really address that issue of let's respond to that pathogen and try to get rid of it. So this shows you just in a little bit more simplistic detail what's happening in the classical pathway. So here we've got a pathogen right here. And this particular pathogen has certain unique molecules on its surface, an antigen. And if your body produces antibodies against that antigen, that's another whole issue we need to talk about after break is how does your body start producing antibodies against these antigens? But if it does, then the antigen-antibody complex that forms on the surface of the cell basically serves as an initiation point or a substrate that allows these complement proteins to start interacting and to form this attack complex, which ultimately results in lysis of the cell. And I showed you this previously, but here's a cell covered with attack complexes, and that cell's a goner. Okay. There are other pathways involved that have other significances. We're just going to briefly mention them. One is the alternate pathway, and this is stimulated by some microbial molecules, particularly endotoxin and glycoproteins. Remember that endotoxin is that lipid A molecule that's part of the LPS layer. And virus particularly have all these glycoproteins on their surface, especially if they're an envelope virus. And those little spikes that kind of stick out of the surface are oftentimes receptors that allow those viruses to attach to the cells and infect them. So those can initiate a complement cascade as well. But it's a different sequence of events. You still form the attack complex, but typically you don't form all of these spin-off products. So this is a more limited response. And then finally, there is a pathway called the lectin pathway. And this is very specialized. Okay? Lectins, are ligands. What's a ligand? Yeah, it's a molecule that attaches to another molecule. Okay? So we're going to see antibodies are going to be considered to be a type of ligand. These particular ligands attach specifically to sugar molecules. And the sugar molecule that most oftentimes attaches to the lectin ligand is mannose. And mannose is kind of an interesting sugar. It's very typically found associated with bacterial cells, and particularly their cell surfaces, but not typically at all associated with eukaryotic cell surfaces. So again, there are these ligands, these lectins floating around, that if they see a mannose molecule on the surface of a cell, they bind to it. And then that can initiate a complement cascade and ultimately lead to the formation of an attack complex. So this is a very powerful method of responding to infectious agents. And in some cases, it doesn't even require the production of antibody to happen, in the case of the alternate pathway or the latent pathway. One of the more fascinating and complicated immune responses that are also part of the second line of defense is the process of inflammation. Have any of you ever suffered from inflammation? Everybody does. Okay. Inflammation is a generalized response to a number of different injuries. It can be a response to an infection. It can be a response to a mechanical injury, a twisting of an ankle, a twisting of a knee. It can be a response to a burn. It can be a response to cancer. And it can be an inappropriate response to things that shouldn't hurt you, but the body has mistakenly identified as being a threat, i.e. allergies. What's the goal of inflammation? Well, there's several goals. One of them is to help recruit immune cells. And that's going to be very important in infection, cancer. Another is to promote healing. Healing can involve initially clot formation, to temporarily seal off a rupture in a vessel, for example. It can later involve cell regeneration and healing of tissue, replacing the damaged tissue. Sometimes scars form, sometimes it's not. Another function of inflammation is to limit your mobility. And that's particularly true in mechanical injuries. It's a sign that something's wrong and you've done something bad, and you shouldn't keep doing it. And the symptoms associated with inflammation oftentimes preclude that kind of additional activity. Now, there's a risk, particularly if you're an athlete, and your injury that is causing inflammation means you can't play or participate. But sometimes it's a response. Get an injection of some type, for example, like a steroid. And what does steroid do? Steroids in general tamp down the immune response and suppress it, including the inflammatory response. Now, sometimes that can be necessary, but other times you're really taking a risk, because if you go out there and you suppress this response and you're already injured, you can injure yourself further. There's two types of inflammatory responses. There's an acute response and a chronic response. And an acute response should be self-limited. You should recover from it. And eventually the symptoms associated with inflammation should subside, particularly as healing occurs and the immune cells do their thing. Chronic inflammation is actually a disease state of itself. The initiating event might have been legitimate, but what's happened is that this immune process doesn't downregulate itself. It doesn't shut itself off, and it just continues. And as more damage is done actually by the inflammatory response, that triggers more inflammation. A lot of these chronic diseases are actually autoimmune disease. Anybody have someone that they know that's older that suffers from chronic rheumatoid arthritis, for example? Pretty nasty disease. It's an autoimmune disease. And the inflammatory response is launched against connective tissue, particularly associated with the joints. You can get very swollen, immobilized joints. And it really is debilitating. That's a chronic state of inflammation. That is a form of, remember I used this term before, immunopathogenesis. That immune-friendly fire. In these particular cases, steroids might be indicated. Excuse me. So what are some of the symptoms of inflammation? Swelling? Sure. Yep. Yeah, let's just start out. This last one. That's a result of these. Happens when you twist an ankle. What are you missing? Bruising is a consequence of inflammation. Pardon me? Or pain. Inflammation has been recognized for a long time. The cardinal signs of inflammation are rubor, tumor, calor, and dolor. Those are Latin words for swelling, redness, heat, and pain. Acute inflammation is supposed to be good. Protective. But the symptoms are not particularly pleasant. One of the things we need to dig into is how and why does inflammation produce these particular symptoms? What's going on here? How can these particular outcomes be part of a process that is protective and good? One of the things that's really affected by the inflammatory process are blood vessels. And one of the things that happens during inflammation is that blood vessels dilate. Another thing that happens during inflammation is that typically blood vessels become more leaky, increase in permeability. Why is this happening? Well, there are a variety of cytokines that are produced during the inflammatory process. And we'll name some of these, but typically many of these cytokines affect blood vessels and cause them to dilate or cause them to become more leaky. What produces these cytokines? Well, neutrophils can produce them. Remember that we said that neutrophils were filled with granules in their cytoplasm. They're granulocytic cells. And under the right conditions, they can degranulate and release these cytokines. The cousin of neutrophils, if you like, mast cells, have the same response. They can degranulate and release cytokines. Mast cells are basically fixed neutrophils that are associated with connective tissue. Another source of cytokines is the blood clotting process. Blood clotting is another complicated cascade event. And again, in an advanced biochem course or medical school course, you probably go through all the interactions of clotting. And yes, one of the big outcomes of the clotting process is this blood serum protein fibrin becomes basically coagulated into this mesh called fibrinogen. And that helps to seal off the vessel, trap bacteria, and things like that. But additionally, some of the clotting proteins that are produced have an effect on blood vessels. The complement cascade we just saw can also spin off these inflammatory mediators that affect blood vessels. So what's going to be the effect if you dilate a blood vessel in terms of blood flow to an area? You're going to get less blood flow or more? You're going to get more blood flow. Is that good? Well, yeah, because there's immune cells circulating around, and so that can increase the movement of blood cells to that particular site of infection. What's that going to contribute to, the increased blood flow? Redness. And heat. As those blood vessels dilate and become more leaky, what's going to happen? We're going to get more diapetesis occurring. And we're going to get more blood cells migrating out into the tissues. Phagocytosis will be occurring. Later, some macrophages might come in and participate in this process as well. And macrophages can start recruiting lymphocytes, so we can start getting some antibody production associated with this. Okay. As the vessels become more leaky, the clotting proteins leak out into the surrounding tissue. Cause blood clotting. Some of the complement proteins leak out into the tissue and become chemotactic substances or opsonins. Well, what's going to happen as fluid starts to leak out into the surrounding tissues. What's going to be the result? Swelling or edema. Okay? You remember what eventually resolves that edema and drains off that fluid? Lymphatic system. Okay. You're not permanently swollen. That extra fluid, in addition to carrying some of these protective molecules, helps to dilute toxins, for example, that the pathogen might produce. As the tissue becomes swollen, it can press on nerve endings. What does that result in? Pain. Okay. So these symptoms associated with this generalized response to all these different kinds of insults, if you like, results in these kind of symptoms, but the symptoms are really a product of primarily cytokines interacting with blood vessels and changing their diameter and changing their permeability. So here's a brief overview of inflammation, and there are several slides that I put in here that are good reviews of kind of this concept right here. So here we've got some type of mechanical injury right here, maybe a splinter of some type, possibly contaminated with bacteria. But the presence of the bacteria is not absolutely necessary to initiate inflammation. You could poke yourself with a sterile something, and you can still get an inflammatory response. Why? Because you're damaging tissue. And damaged tissue, like mast cells, can start releasing cytokines. Those particular cytokines can have an effect on the permeability of the blood vessels, ultimately increasing movements of those phagocytic cells out into the surrounding tissue, and in this case, dealing with the infectious agent by phagocytosis. This talks a little bit about the changes in permeability that occur to the cell walls. Capillaries are basically one cell layer thick. They're a bunch of endothelial cells stuck together to form a tube-like structure. And typically you only get a small amount of fluid leakage out of the circulatory system into the tissues. So the white blood cells are usually kind of confined to the circulatory system. But in response to inflammatory substances, these various cytokines, permeability can change, dilation can loosen up those junctions between the cells, and now we can get some diapetesis occurring. That's going to start bringing more of those protective cells to the site of infection. This is another thing. This talks a little bit about some of the chemical mediators or cytokines that are released as a result of the inflammatory response. Some of these you've heard of before, probably. You've all heard of histamines before, right? Well, histamines are involved in the part of the inflammatory response that involves allergy. So what is one of the treatments for allergy? Antihistamines, okay? Molecules that block histamine release. Some other things that are produced include prostaglandins, leukotrienes, and prostaglandins are going to be kind of important. Prostaglandins can do a couple of things. They can stimulate inflammation. In part by causing blood vessels to dilate. And they can stimulate fever. We said that the pain associated with inflammation is oftentimes the result of the leakage of fluid into the surrounding tissues. And that would be promoted by prostaglandins, okay, more leakage. What is something that you can take fairly routinely that will both decrease pain and fever? Ibuprofen, aspirin. Those particular drugs work by inhibiting prostaglandin synthesis. there is an enzyme called the COX-2 enzyme, which is involved in prostaglandin biosynthesis. And those NSAIDs, you know what NSAID stands for? Those are the non-steroidal anti-inflammatory substances, like ibuprofen and aspirin, are inhibiting this COX-2 enzyme, decreasing prostaglandin production, which then has an effect on your pain and your fever.
These drugs, in some way.
They have an effect on the brain in that they affect the hypothalamus, which is the temperature regulating center. But NSAIDs do not kind of numb a section of the brain responsible for pain. No. Dampen inflammation, which is causing the pain. Okay? Leukotrienes, you might have heard some about leukotrienes if you ever see ads on TV for various drugs. Leukotrienes are also involved in asthma, which is kind of the inflammatory response of the lungs. So I know some of the drugs are advertised as being able to depress leukotrienes. But again, under the response of all of these cytokines, we get this alteration of blood vessel permeability, and that results in the movement of these protective cells and molecules to that site of infection, ultimately resulting, hopefully, in healing. This is just kind of a flow diagram. This is another way to represent the changes that occur during the process of inflammation. This particular table just shows you what some of the players are in the inflammatory response in terms of cytokines. There are various vasodilating chemicals, histamine, serotonin. Bradykinins are produced during the blood clotting process. I mentioned prostaglandins. There's various chemotactic factors that are involved in inflammation. Fibrin is part of the clotting process. Mast cells produce various chemotactic factors. There are actually some bacterial compounds that are chemotactic in nature. And then there's some substances that have both vasodilating and chemotactic effects. This is another slide that gives you kind of a rundown of some of the major cytokines that are involved in the inflammatory response. It's a complicated response. So there are a couple more slides that kind of puts together inflammation along with diapetesis. I'm not going to go through all that right now. Fever is sometimes associated with inflammation, but not always. Fever is initiated when substances called pyrogens have an effect on the hypothalamus. These particular compounds cause the hypothalamus to produce prostaglandin. And prostaglandin is a molecule that regulates the temperature sensor in your brain. Okay? Normally your thermostat is set to approximately 98.6 degrees Fahrenheit. But in response to infection, prostaglandin synthesis can be increased. That affects the hypothalamus, and it raises the temperature sensor. So now we have a fever. What would be the advantage of jacking up your temperature system? Yeah, I mean, if you put your bacteria, your unknown, in the wrong incubator, you might kill it, right? Organisms have an optimal temperature, a maximum temperature, a minimum temperature. But there are other benefits to fever as well. In general, fever increases the activity of immune cells. It can cause them to be more proficient in producing cytokines. It can cause the bone marrow to increase production of blood cells, particularly white blood cells. It can have an effect on the iron availability in the blood. Remember, iron is a limiting factor to bacterial growth. So all kinds of benefits from fever. Why are you getting hot? Well, the hypothalamus is basically sending nerve signals that cause you to shiver, and that muscular contraction of shivering generates heat. It causes blood vessel dilation. And basically, it will persist as long as there are enough pyrogens around to stimulate enough prostaglandin to keep the temperature sensor set artificially high. What happens when the infection starts to resolve itself? Pyrogen levels drop, prostaglandin levels drop, and the temperature set point reduces itself back down to normal. And what happens? Well, now your body's hot. So the cooling mechanisms start to be activated, like sweating. And so that's oftentimes referred to as the fever breaking, and that's usually indicated as a good sign, because now your body is going, I'm too hot. My temperature sensor says I should be now 98.6 degrees again. Therefore, I'm going to do things to cool down. Okay? So the question becomes, if fever is beneficial, why do we take NSAIDs, for example, to suppress fever? Is that a good strategy? What's it depend upon? How high the fever is. Okay? If you get above about 104, that's problematic. And there, the negative effects of the high temperature on your body cells outweighs the benefit of trying to kill the pathogens. It's more an immediate problem to preserve your body cells. So you need to bring that fever down. Alcohol baths, something to lower the temperature. But these low-grade to moderate fevers, while they're uncomfortable, have a beneficial effect. And so next time you're suffering from a fever, you might want to, number one, take your temperature and see how bad it is. But number two, resist that temptation to try to suppress the fever, because you're suppressing one of the parts of your defense system. So fever management is important. Then there's just some summary slides of the non-specific components of the first and second line of defense. So here's where we are right now. Remember, the goal is to kind of understand this theatrical production that we call the immune system. We've looked at the setting of the immune system, the theater if you like. We've looked at some of the players. We've looked at some of the processes. But what we really want to dig into now is that specific third line of defense, that acquired part of the defense that involves memory and specific antibodies. So we're going to have to dig a little bit more deeply into those particular actors that are responsible for antibody production, the lymphocytes. To do that, we're going to have to dig into some additional details. And one of the details will be cell surface receptor molecules on immune cells. So we're going to talk about MHC1s and MHC2s. We're going to have to talk about antibodies. What are they? What are the different types? We're going to have to talk about antigens, the molecules that interact with antibodies. And once we do that, then we can put all this together and talk about the coordinated immune response, which involves both the cell-mediated immunity, the T lymphocyte part, and the humoral immunity, the B lymphocyte part. So again, I can't over-iterate the fact that this is stuff you really do need to keep up with. And if you are kind of struggling with some of the terminology and things that we talked about now, then that's going to make understanding when we try to put the whole thing together more difficult. So really try to keep up with this particular information. Get a 5-minute break today. So have a great Thanksgiving. Remember, don't leave things in the incubator. Either throw them away or put them in the refrigerator. Have a good break. So, what's your interest?
So that's good as well. I didn't know what you meant.
Okay. What makes you want to be a PA? Okay. Have you ever spent any time with a PA or done any shadowing? Or?
Yeah,
I think most people, I
the actual,
Those are just basically.
they're just fans, so after
yeah,
surgery, actually.
How old is high school? Just running short
so I just sat down and we talked.
Well, and you can have been blood too, because you kind of like run like
Yeah.
Well, there's certainly a well-marked.
Yeah, it was like this.
And I think what attracts people to PA too is the fact that being an MD can kind of be considered.
Yeah.
And I think PA, some people feel like it's a little bit more of a balanced life choice.
Yeah.
You don't have to have malpractice insurance. So I think it's a nice choice. So were you seeing?
Oh, yes.
Where was it?
Something.
Okay. So where are you in your process and college decisions?
It's been here at Anderson.
Oh, okay. So have you similar to classroom business and things to Anderson?
I did an overnight, but we didn't do any classroom stuff, so that kind of worried me. So
Are you doing it overnight here?
I already did.
Oh, you did?
Last night. Yeah.
Okay, just take it for you.
Yeah.
Hopefully we didn't
And she
scare
didn't
her
scare
off.
you away?
No.
1 o'clock in the morning, and it sounds like tribal drums are playing from the other room. I'm like,
We were good girls last night.
Nice.
None of us had a lot of homework today, which is like a first for like my roommates and I. Just all of like we were just like good girls last night, which is nice. Yeah.
So anything you would want to do?
Do we have to
besides,
check that one?
Study science?
like, are we usually that in there longer than 24 hours? I like softball. That would
Okay.
that'd be rough though, with like PA and biology. So.
We have a lot of students who do.
Really?
Yeah, we have a lot of varsity athletes, both men and women. And, it's our science people are something more involved in. Golfer over here, and
Yep.
a lot of our students are really pretty involved, and that's one of the reasons why they like the small school.
Yeah.
You can do that. You just have to be realistic and say,
It won't be
okay, I'm not
Yeah,
going to
much
pay $1,000 when
I'll help you 1 second. I just gotta
Yeah,
play
let's
softball. Yeah.
but it's certainly possible, and in some respects, I think it's good to have that particular kind of extracurricular activity.
Yeah.
The other thing that is really important in a lot of programs, we're still working on ours. We expect to get them running by 2019. One of the things we don't know is how much patient care, contact hours we will require, there'll be some requirement. So we're really trying to work on providing some opportunities for our students. We have a big retirement home just by campus, Memphis, also Memphis. And one of the things that we're going to work with them on is there's a certification you can get called the CNA, Certified Nursing Assistant. And CNA is a great way to do patient contact hours. You're also paid. And the proximity to campus makes it really nice because you can actually work around and work some hours during the semester, banking up your patient care hours and get a little bit of extra money. So that's going to be considered too, which are going to be opportunities for you to get those patient contact hours. Anything else you're curious about while I'm sitting here? So is this the only class you're sitting in, or are you taking groups to someplace else?
We're going to like 201 next.
Oh, okay.
So,
Who's teaching you 201?
Dr. Cruz Foley.
Okay.
She's pretty much awesome.
So what type of instrument?
My volleyball coach.
Okay.
She went here, and then I work at a golf course, and I guess he's at Wabash now, but Alan Hill, he worked in.
Oh, yeah.
Yeah. He came golfing, and he talked to me like for like an I'm not at Franklin anymore. He's like, but
But you can't come to Wabash, so.
Yeah.
so might as well go to
he's like, if
Franklin.
you're still interested, he goes, I still have connections there. And I was like, all right.
Thank
It's
you.
awesome. It is funny. He's a really nice guy.
Ready?
Yeah.
Okay, well, it's nice to meet you.
Nice to meet you too.
And if you have any questions, Corey can get hold of you.
See you, everybody.
See ya, have a good day.
Thank you.
No, it's also in there.
I must admit, I do like inflammation.
I really like this section.
Immune system stuff, yeah. I mean, it's just so interrelated and complicated and feedback, but I mean, once you, it takes time to wrap your head around it and kind
Yeah.
of get a sense of how it all fits
I like
together.
it more like processes though, like this, and rather than like medications and stuff. I like knowing what things are doing.
Yeah.